Hi, welcome to the BYU-Idaho Tutoring Center videos. Today we're going to be talking about point mutations. So to start off, a point mutation is a chemical change in a single base pair of a gene. And so there's two types that we usually talk about. There's substitutions and insertion deletions. And so as we go through these today, I'm going to be using an example that I've written at the top of the board. So if this is our original strand of DNA right here, and this is our template strand, then if this is our template strand, this would be the mRNA strand that formed, and this would be the protein or polypeptide or sequence of amino acids that would form from that mRNA. So this is transcription and this is translation going on. And so what we're going to be doing is we're going to be messing with this template strand or this original DNA strand, and we're going to see how it affects the rest of this. Because we have a better understanding of how DNA forms a protein, we're going to be able to understand these changes. So to start off, we're going to be talking about substitutions. So in substitutions, we just trade out an amino acid for a different amino acid. Sorry, not an amino acid. We change out a nucleotide for a different nucleotide in our DNA, and thus it changes the one that it's paired with as well. So first, we're going to look at this. If we change out our C for a T, and so then our G is going to be traded out for an A, then we're going to see this as our new mRNA sequence. So now, instead of having a C in that position, we're going to have a U in this position. But you notice when we translate our mRNA into a protein, we're going to have the same sequence of amino acids. And that's because of the redundancy in the genetic code. GGU and GGC both code for glycine. So when this occurs, when we don't have a change in our polypeptide or our protein as a result of a change in DNA, we call this a silent mutation because it didn't affect anything. However, this is not always the case. In this next example, we change a G to an A, which then results in the C being changed to a T. And so as a result, our mRNA now has an AGC instead of a GGC. And as a result, instead of forming glycine, we're going to form this serine instead. So now we have this different amino acid in this spot. So that's called the missense mutation, when we do affect our amino acid sequence. And sometimes this isn't going to have a big effect. A lot of times we'll still be able to fold our protein and it's still going to be pretty functional. However, if this amino acid that we change is in a key spot or location, especially if it's in a location that helps bind a substrate, then we can have a lot of problems. And so an example of this is sickle cell disease. We have one base pair substitution, which results in one change in amino acid, and that amino acid completely changes the protein and results, or the hemoglobin, which changes the shape of the red blood cell, which results in a big problem and sickle cell disease. And so it really just depends on where this amino acid is. And this can also be influential for beneficial mutations because if we change this amino acid in a certain location, it might add on a new important function to the protein or better ability. And so if we have that better ability, the organism may have some advantage over those that didn't have this mutation. And so it really just depends on where we have this missense mutation to determine whether it's going to cause problems or whether it's going to be beneficial or whether it's not really going to make a difference. Last one is if we change this A right here to a T, and thus the T is going to change to an A, and then our mRNA sequence is going to have a U in this spot instead of an A, we're going to have a premature stop codon. And so instead of forming this longer amino acid sequence, now we have this really short one because we had our stop codon instead of amino acid codon. And so when this happens, we usually end up forming a shorter polypeptide, and as a result, it usually is not functioning because it can't fold properly. If this premature stop codon is pretty at the end, then sometimes it's not going to have as big of a impact on our amino acid. But in general, rule of thumb, these nonsense mutations, that's what they're called, are going to result in a non-functioning protein. Okay, so now we're going to move on to insertion deletion. And these ones cause a lot more problems than substitutions, or they tend to cause a lot more problems. And so what we do is we either take out a nucleic acid where we add in a nucleic acid. I'm sorry if I've been saying amino acid when I meant to be saying nucleic acid a couple times back here. So what happens is, in this example right here, we added this T. So before it was ATGAA, and now it's ATGTAA. And so when we add that, everything is shifted over. The 3 codon reading frame is moved over. And so in this example, instead of moving on to form lysine, we actually have a premature stop codon. So this is another nonsense mutation, but it's a result of a base pair insertion. And the same type of thing can happen with deletions. If we get rid of a T right here, so we had 3 T's, we got rid of one of those T's, it's going to cause everything to shift over. So instead of being AUG, AAG, UUU, now it's UUG. And as a result, this shifts this over as well. So now we don't have our GGC, we have GCU, because everything was shifted over. That's why it's called a frame shift. And as a result, we're going to form the 2 first normal amino acids, but then we're going to form an amino acid we didn't expect, and then we're going to have skipped our stop codon because we changed it, the reading frame. And so we're going to keep forming these amino acids when we're not supposed to. We're going to form some really weird polypeptide, and it's going to form some really weird protein that we don't want. So you can see how just changing, we're adding or deleting one base pair is going to completely shift everything over. So unless this happens at the very end, it probably is going to have a big effect on our polypeptide. And if it does happen at the very end, it could cause some problems because it could get rid of a stop codon that we needed. So the only way that this isn't going to cause a ton of problems is we have a 3 base pair insertion or deletion. Because remember, the codons read in 3s. And so if we get rid of a total of 3 or add a total of 3, then we're just going to be adding or getting rid of an amino acid. So in this example, if we get rid of the AAG that was up here, then all we're going to be doing is getting rid of this amino acid lysine. And everything's going to be virtually the same. And so this polypeptide might be abnormal or not be as functional as this one, but it's not going to be as big of a change as some of these other base pair insertion deletions. So just remember, if we have anything that's on a scale that's not divisible by 3, so if we have a 6 base pair insertion or deletion or 9, it's not going to have as big of effect if we have 1 or 2. And so just be aware of that. So the last thing I want to touch on is just the mutagens, or how we have these nucleic acid changes. A lot of it can just be errors with DNA replication. And so if we just have mistakes with our polymerases. Another thing is sometimes we just have spontaneous mutations. We don't really understand them. They just kind of happen randomly with everything that's going on in the cell. We have a harder time tracking these. However, keep in mind that with all of these mutations, only about 2% of our DNA actually codes for genes. So if we have quite a few mutations in non-coding DNA, then it's not going to cause some problems. And the last thing is we have these things that are actually called mutagens, such as X-rays or UV light or different chemicals. And they just alter our DNA. They can break up dimers where they're supposed to be, or different chemicals can come in and take the spot of different nucleic acids and just cause other problems like that. So just be aware that just different chemicals and light exposures, and then just error with replication all cause these changes, which then will ultimately affect the protein that we produce. That is all for today. Thank you so much for watching. I hope you understand point mutations a little bit better, especially substitutions, insertion deletions, frame shift, and how they all will ultimately affect the protein. Keep in mind that it's going to be different in every case. Not every time we have a 1 base pair insertion, that's not going to mean we're always going to have a nonsense, or not every time we have 1 base pair deletion isn't always going to mean we're going to have a run-on polypeptide. It just really depends on the situation. So whenever you get a problem like this, really work it out, write out the mRNA, then follow the codons and the anti-codons to figure out what amino acids we will be producing, and see how that changes from the original. Again, thank you so much for watching, and have a fantastic day.